Hello students, my name is Dr. Asha Lucky and today we are going to discuss NEET 2018 question paper. I will be dealing with the biology section and specifically we are going to look for the botany questions. So we are going to start with the question number 91. The question number 91 goes like oxygen is not produced during photosynthesis. Options are cycles, nostoc, green sulfur bacteria and chara. So for this question, we should know about the photosynthesis. We know that photosynthesis, it is a process where the food molecule, that is C6H12O6, is synthesized. So during the synthesis of the food, we require two important major raw material. One is the carbon dioxide and second one is the hydrogen source. We require hydrogen source. So this hydrogen source, it can be either water molecule or it can be any other hydrogen source. For example, if we say hydrogen sulfide. If water is used as a source of hydrogen, we are going to get the byproduct, that is oxygen. And if you are going to take hydrogen sulfide as a source of hydrogen, then sulfur is going to be our byproduct. Now coming back to the options. So here we can see the first option is cycas. So cycas, it is a plant which belongs to gymnosperm. Gymnosperm, it is a developed plant. तो यहाँ पे जो photosynthesis होगा वहाँ पे oxygen produce होगा, तो इसको हम oxygenic photosynthesis कहेंगे. The second one is the nostoc. Nostoc, it is otherwise known as blue-green algae. Blue-green algae is considered to be the first organism which is going to produce the oxygen during photosynthesis. Then next coming to the third option, that is green sulfur bacteria. Bacteria, it is a prokaryotic cell. ये प्रोकैरियोटिक सेल है. प्रोकैरियोटिक सेल में ऑक्सीजन प्रोड्यूस नहीं होता एंड हियर द नेम ऑल्सो इंडिकेट्स ग्रीन सल्फर बैक्टीरिया, दैट मीन्स द बाई प्रोडक्ट हियर प्रोड्यूसेस सल्फर. जब सल्फर बाई प्रोडक्ट हो रहा है तो ऑक्सीजन बाई प्रोडक्ट नहीं होगा. तो नाउ कमिंग टू द लास्ट वन, दैट इज विल सी दैट वन ऑल्सो, दैट इज द चारा. इट इज एन algae. ये भी एक algae है. यहाँ पे भी ऑक्सीजनिक फोटोसिंथेसिस होगा. सो द ऑप्शन अंडर दिस वन इज ग्रीन सल्फर बैक्टीरिया, जो कि प्रोकैरियोटिक सेल है. यहाँ पे नॉन ऑक्सीजनिक फोटोसिंथेसिस होगा, सो ऑक्सीजन इज नॉट प्रोड्यूस्ड ड्यूरिंग दैट प्रोसेस. द नेक्स्ट क्वेश्चन इज double fertilization is, the options are fusion of two male gametes with one egg cell, fusion of one male gamete with two polar nuclei, fusion of two male gametes of a pollen tube with two different eggs, syngamy and triple fusion. So for this we should know about the double fertilization. तो ये double fertilization जो है ये it is an important characteristic features of angiosperms. तो angiosperms में जो fertilization होता है वो double fertilization होता है. We will look for that कि angiosperm में fertilization कैसे होता है. So उसके लिए we have taken this diagram. So इस diagram में we can see this is our embryo sac. ये है हमारा embryo sac. इस embryo sac में we are having totally 8 nucleus. हमारे पास 8 nucleus है and it is 7 cells है. The arrangement of 7 cells, the 3 cells are at this polar end and the 3 cells are at this polar end, and the one cell but 2 nucleated one cell is present at the center. This is known as the polar nuclei, या फिर हम उसको secondary nucleus भी कह सकते हैं. We can call it as secondary nucleus. So now what is happening here? So during the fertilization, the two male gametes are going to fertilize the two cells. One cell is our egg cell. Egg cell यहाँ पे fertilize होगा and that is going to result into zygote. हमें zygote मिलेगा. Zygote will develop into embryo and embryo is going to germinate to form the plant, right? Now the second fertilization is with the secondary nucleus. यहाँ पे already 2 nuclei present है, one more male gamete is going to fuse with this one and this result into a structure जिसको हम कहते हैं एंडोस्पर्म. एंडोस्पर्म ये 3n स्ट्रक्चर है, जाइगोट 2n स्ट्रक्चर है. क्योंकि ये 3n स्ट्रक्चर है हम इसको कहेंगे ट्रिपल fusion because 3 nucleus are fusing here, 2 nucleus of the secondary nuclei and 1 from the male gamete. तो triple fusion. and यहाँ पे the egg cell is fusing with the male gamete. यहाँ पे egg cell male gamete के साथ fuse कर रहा है तो ये हो गया syngamy. इसको हम क्या कहेंगे? syngamy that results into zygote. तो triple fusion and syngamy that results into double fertilization. Coming to the options here, तो यहाँ पे जो ऑप्शन फिट बैठता है डबल फर्टिलाइजेशन के लिए वो है syngamy and triple fusion. Syngamy results into zygote and triple fusion results into endosperm. So हमारा जो सही आंसर है वो है फोर्थ ऑप्शन, that is syngamy and triple fusion. Next question is, which one of the following plant shows a very close relationship with the species of moth, where none of the two can complete its life cycle without the other? And option goes banana, yucca, hydrilla and viola. So यहाँ पे जो क्वेश्चन दिया गया है, so they are asking about the relationship with the species of moth. जब हम इस मॉथ स्पीसीज को देखते हैं तो वी ऑलवेज से दैट इट इज़ इन अ सिम्बायोटिक रिलेशनशिप विद द प्लांट. सिम्बायोटिक रिलेशनशिप. ये ऑब्लिगेटेड रिलेशनशिप होता है. सो इन दिस केस द मॉथ रिक्वायर्स द हेल्प ऑफ द प्लांट टू कंप्लीट इट्स लाइफ साइकिल. सिमिलरली द प्लांट इज ऑल्सो इन अ सिम्बायोटिक रिलेशनशिप, तो इट ऑल्सो रिक्वायर्स the help of the moth to complete its life cycle. So we have a diagram, we have a figure for that one. See, this is the figure here. We can see the moth and the flower, and this flower, it is the yucca flower. तो हमारा जो ऑप्शन बैठेगा वो होगा yucca. तो yucca, it is a plant in which the moth species are going to hatch their eggs. तो एग सेल्स जो हैं वो हैच कहाँ पे होते हैं? इन द फ्लावर ऑफ द यूका. अलॉन्ग विद दैट इट ऑल्सो ईट द सीड्स टू गेट द न्यूट्रिशन. सिवाय यूका सीड्स के मॉथ कोई भी और सीड्स को कंज्यूम नहीं करते. एंड अपार्ट फ्रॉम दैट इफ़ वी टॉक अबाउट द यूका, यूका में जो पॉलिनेशन होता है वो सिर्फ और सिर्फ कौन से ऑर्गेनिज्म कर सकते हैं? द मॉथ. नन अदर ऑफ द ऑर्गेनिज्म कैन डू दैट प्रोसेस. So that's why we call this as obligatory symbiotic relationship, जहाँ पे yucca depends upon the moth and the moth depends upon the yucca. So हमारा जो आंसर है यहाँ पे वो होगा yucca. The banana, the hydrilla and the vulva are the wrong options for this. Next question is, pollen grains can be stored for several years in a liquid nitrogen having a temperature of, options are minus 196 degree centigrade, minus 80 degree centigrade, minus 120 degree centigrade and minus 160 degree centigrade. So here first we should know that when we are preserving any living organism or any living component, हम liquid nitrogen को prefer करते हैं. So this method of using liquid nitrogen and preserving that at a very low temperature, उसको हम कहते हैं cryopreservation. So that method is known as cryopreservation, the preservation at very low temperature. तो why we are preferring the liquid nitrogen? क्योंकि liquid nitrogen का जो boiling point है या boiling temperature है वो बहुत low है. उसका जो boiling temperature है वो बहुत ज़्यादा low है. तो extremely low temperature is preferred to preserve the पोलन ग्रेन्स. पोलन ग्रेन्स को हम उस टेम्परेचर पे स्टोर कर सकते हैं एंड दैट टेम्परेचर इज -196 डिग्री सेंटीग्रेड. तो एक्सट्रीमली लो टेम्परेचर, एंड लिक्विड नाइट्रोजन का जो बॉइलिंग टेम्परेचर है वो -196 डिग्री सेंटीग्रेड है। तो एट दिस टेम्परेचर वी कैन प्रिजर्व और वी कैन स्टोर द पोलन ग्रेन्स। तो हमारा जो ऑप्शन बैठेगा वो है -196 डिग्री सेंटीग्रेड। Next, which of the following elements is responsible for maintaining turgor in cell? Options are potassium, sodium, magnesium, calcium. So here इस क्वेश्चन में वो टर्जिडिटी के बारे में बात कर रहे हैं, दे आर आस्किंग अबाउट द टर्जिडिटी। तो टर्गल सेल्स और टर्जिडिटी इन सेल इज नथिंग बट द प्रेशर ऑफ वाटर इनसाइड द सेल। सेल के अंदर जो प्रेशर डेवलप हो रहा है एंड दैट प्रेशर इज डेवलप बिकॉज ऑफ द वाटर, तो उसको हम कहते हैं टर्जिड सेल। सो सेल के अंदर अगर वाटर का प्रेशर डेवलप होना है तो देन देयर शुड बी अ प्रोसेस जिसको हम कहते हैं एंडोस्मोसिस। एंडोस्मोसिस, द एंट्री ऑफ वाटर inside the cell. This is going to generate the pressure on the cell and the cell is known as turgid cell. उस सेल को हम कहेंगे turgid cell. तो इस एंडोस्मोसिस के लिए there is a ion responsible and the ion which is responsible for that one is the potassium ion, that is K plus ion. तो K plus ion है जो एंडोस्मोसिस के लिए रिस्पॉन्सिबल है and as a result that is also responsible for the turgid cell. It is an important mechanism which is used during the opening and closing of stomata. वहाँ पे potassium is going to play a major role. तो हमारा जो आंसर है यहाँ पे वो है potassium. Our next question is, what is the role of NAD+ in cellular respiration? The options are, it is a nucleotide source for ATP synthesis, it functions as an electron carrier, it functions as an enzyme, it is the final electron acceptor for anaerobic respiration. तो यहाँ पे they have asked about the cellular respiration. तो ये क्वेश्चन आया है हमारा चैप्टर respiration से। तो we know that the respiration is the process of synthesis of ATP. यहाँ पे ATP molecule synthesize हो रहा है। So for the synthesis of ATP, the food molecule is broken down, that is the complex food is broken down into simple food to produce an energy, that is ATP. तो यहाँ पे during that process the NAD+ is used. तो NAD+ क्या करेगा? It is going to act as an electron carrier. वो electrons को carry करेगा, the electrons which are released during the breakdown of the food molecule, and that electrons are later used for the synthesis of ATP तो through the electron transport system. तो यहाँ पे जो first option है nucleotide source for ATP synthesis, it is not a source for ATP synthesis. It is not acting as an enzyme, neither it is used as a final electron acceptor in anaerobic respiration. तो हमारा जो सही ऑप्शन है वो है it functions as electron carrier. ये इलेक्ट्रॉन को कैरी करता है which is later used in the synthesis of ATP. Next question is, in which of the following forms is iron absorbed by plants? The options are free elements, ferrous, ferric, both ferric and ferrous. ये जो क्वेश्चन है थोड़ा सा कॉम्प्लिकेटेड क्वेश्चंस हैं। इसको हम कॉम्प्लिकेटेड क्वेश्चंस इसीलिए कह रहे हैं बिकॉज when we look for the actual mechanism, तो प्लांट में जो आयरन एब्जॉर्प्शन होता है that is in Fe+2 and Fe+3, it means both ferric and ferrous, इन दोनों के फॉर्म में the iron is absorbed. But when we specifically look for the NCERT, तो जब हम NCERT के बेस में जाते हैं then iron is absorbed in the form of ferric ion. So NCERT के हिसाब से जाना पड़ेगा क्योंकि ये हमारा नीट का क्वेश्चन है। तो इफ़ यू गो विद द NCERT और थर्ड ऑप्शन, अदरवाइज द प्लांट एब्जॉर्ब्स द आयरन इन बोथ फेरिक एंड फेरस फॉर्म. The next question is, which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Options are lambda-phage, TI plasmid, retrovirus and PBR322. तो सबसे पहले हम लोग देख लेते हैं वेक्टर फॉर इंट्रोड्यूसिंग डीएनए फ्रैगमेंट. तो ये जो वेक्टर्स हैं, these vectors are used as a vehicle टू ट्रांसफ़र द डीएनए फ्रैगमेंट। डीएनए फ्रैगमेंट को ट्रांसफ़र करने के लिए हम वेक्टर्स यूज़ करते हैं। वी हैव डिफरेंट टाइप्स ऑफ वेक्टर्स डिपेंडिंग अपॉन द होस्ट सेल। किस सेल के अंदर आपको वेक्टर या किस सेल के अंदर आपको डीएनए फ्रैगमेंट को ट्रांसफ़र करना है, उसके बेसिस पे हमारे पास डिफरेंट वेक्टर्स हैं। लाइक फॉर एग्जाम्पल, इफ़ योर होस्ट सेल इज़ बैक्टीरिया, अगर आपका होस्ट सेल बैक्टीरिया है तो हम लोग यूज़ कर सकते हैं बैक्टीरियोफेज। हम बैक्टीरियोफेज को वेक्टर यूज़ करेंगे, और यू कैन ऑल्सो यूज़ द प्लाज्मिड। द मोस्ट कॉमनली यूज्ड प्लाज्मिड इज PBr322। तो मतलब अगर आपको बैक्टीरिया में डीएनए को ट्रांसफ़र करना है तो आपके पास है बैक्टीरियोफेज एंड प्लाज्मिड, दैट इज PBr322। And if you want to transfer the DNA into plant cell, plant cell में अगर आपको ट्रांसफ़र करना है तो most widely used vector is TI plasmid, or we have one more, that is RI plasmid, tumor inducing plasmid and root inducing plasmid, that is TI plasmid and RI plasmid। बट अगर जब आपको एनिमल के अंदर आपको ट्रांसफ़र करना है तो यू हैव टू यूज़ द एनिमल वायरस। हम एनिमल वायरस को यूज़ करते हैं। द मोस्ट वाइडली यूज्ड एनिमल वायरस इज़ रिट्रोवायरस। एंड क्योंकि हमें यहाँ पे ह्यूमन लिम्फोसाइट के अंदर डीएनए को ट्रांसफ़र करना है, सो ऑब्वियसली वी आर गोइंग टू यूज़ रिट्रोवायरस, बिकॉज लैम्डा फेज इट इज़ यूज्ड फॉर बैक्टीरिया, TI plasmid it is used for plant, and PBr322 it is used for plant। एंड हमारा जो सही आंसर है, ह्यूमन लिम्फोसाइट के अंदर अगर आपको जीन ट्रांसफ़र करना है तो यू हैव टू यूज़ अ वेक्टर, दैट इज रिट्रोवायरस। Question is, use of bioresources by multinational companies and organization without authorization from the concerned country and its people is called. Options are biodegradation, second one is biopiracy, the third one is bio-infringement, and fourth one is bio-exploitation. So here the question is that multinational companies and organization, what they are doing, they are using the bioresources without the consent of the कंट्री। ऑथराइज्ड कंट्री को बिना बताए अगर बायो रिसोर्सेज को यूज़ करते हैं तो तब हम उसको क्या कहेंगे। हमारे पास ऑप्शंस है बायोडिग्रेडेशन। तो बायोडिग्रेडेशन इट इज़ नथिंग बट द ब्रेकडाउन ऑफ कॉम्प्लेक्स मॉलिक्यूल्स इनटू सिंपल मॉलिक्यूल्स बाय माइक्रो ऑर्गेनिज्म, तो यहाँ पे ये ऑप्शन फिट नहीं बैठता। द सेकेंड ऑप्शन इज़ बायोपायरेसी। बायोपायरेसी को हम यूजुअली कहते हैं लीगल थेफ्ट। लीगल थेफ्ट मतलब बिना ऑथराइज्ड पर्सन को कंसर्न किए, इफ एनीथिंग इट इज़ यूज्ड बाय सम पर्सन, then it is known as bio-theft, and that is also known as biopiracy. We'll go for the third one also, bio-infringement. It is nothing but breaking the laws, violating the law. law को break करना उसको हम कहते हैं bio-infringement, that is breaking the law। law को break करना या law को violate करना। And next one is bio-exploitation। bio-exploitation that is the excess use of any resource को हम क्या कहते हैं, bio-exploitation। तो excess utilization को हम we are going to call it as the bio-exploitation। जो हमारा सही ऑप्शन बैठता है वो है biopiracy, that is a legal theft without the consent of the authorized person if bioresources are used by any multinational companies or organization. So that option is biopiracy. The next question is, in India, the organization responsible for assessing the safety of introducing genetically modified organism for public use, GMO, genetically modified organism for public use, तो कौन देता है, who is giving the approval for releasing the GMO for the public use? The first option is research committee on genetic engineering। तो रिसर्च कमिटी what it is going to do, ये रिसर्च के लिए अप्रूवल देता है but not for the public use। The second one is Council of Scientific and Industrial Research, again यहाँ पे it is not, again it is for the research purpose. The Indian Council of Medical Research, ये भी नहीं है, बट जो जेनेटिक इंजीनियरिंग को पब्लिक यूज के लिए अप्रूवल देता है दैट इज द जेनेटिक इंजीनियरिंग अप्रीशियल कमिटी. ये आपके NCERT में एज यूजुअल सेंटेंस है, दैट इज GEAC. दे आर रिस्पॉन्सिबल फॉर अप्रूवल ऑफ द GMO फॉर द पब्लिक यूज. पब्लिक यूज के लिए GMO के रिलीज के लिए आपको इनसे परमिशन या इनसे अप्रूवल लेना होता है. So the option is the fourth one, that is Genetically Engineering Appraisal Committee, GEAC. So the next question is again from the biotechnology, that is the correct order of steps in PCR, that is polymerase chain reaction. तो polymerase chain reaction जो है it is in vitro DNA synthesis. ये क्या है हमारा इन विट्रो डीएनए सिंथेसिस. इसको हम एम्प्लीफिकेशन ऑफ डीएनए भी कहते हैं, वेयर द डीएनए इज एम्प्लीफाइड. तो व्हाट आर द स्टेप्स इन्वॉल्व इन दिस इन विट्रो डीएनए सिंथेसिस और एम्प्लीफिकेशन ऑफ द डीएनए. इफ दिस इज आवर डबल स्टैंडर्ड डीएनए, ये है हमारा डबल स्टैंडर्ड डीएनए. तो फॉर सिंथेसाइजिंग व्हाट वी हैव टू डू, वी हैव टू कन्वर्ट दैट इनटू अ टेम्पलेट स्टैंड. तो ये हो गया हमारा टेम्पलेट स्टैंड. तो टेम्पलेट स्टैंड में कन्वर्ट करने के लिए वी हैव टू ब्रेक द हाइड्रोजन बॉन्ड एंड उसको हम कहते हैं डीनेचुरेशन. दैट प्रोसेस इज नोन एज डीनेचुरेशन, दैट इज डन बाय incubating this DNA molecules at high temperature. तो अब अराउंड 92 to 94 डिग्री सेंटीग्रेड तक हम उसको इंक्यूबेट करेंगे. द हाइड्रोजन बॉन्ड विल ब्रेक एंड यू आर गोइंग टू गेट द टेम्पलेट स्टैंड. तो अब टेम्पलेट स्टैंड जब आप करेंगे, सो द नेक्स्ट स्टेप व्हाट वी हैव टू डू, वी हैव टू मेक द प्राइमर्स टू बाइंड टू दिस डीएनए फ्रैगमेंट्स एट अ स्पेसिफिक साइट. तो यहाँ पे क्या बाइंड हो रहे हैं, प्राइमर्स बाइंड हो रहे हैं. सो दिस बाइंडिंग अप ऑफ द प्राइमर इज नोन एज annealing. So binding means annealing. So this is our second step. And once the annealing is done, then we have to go for the third step. हमें third step के लिए जाना है. तो third step में क्या होगा, third step में synthesis होगा. तो synthesis, how the synthesis will take place? Like this, a DNA strand will be synthesized. So single DNA molecule is converted to 2 DNA molecule, and this is by a process called as extinction. This is the extinction. तो 3 step है, denaturation, annealing, extinction. We'll look for the option now. So first option दिया है denaturation, extinction, annealing. नहीं, annealing second होना चाहिए, तो ये सही नहीं है. The second option is annealing, extension, denaturation. No way, पहले denaturation होना चाहिए. The third one is extension, denaturation, annealing. No. And the fourth option, denaturation, annealing and extension. This perfectly fit with the polymerase chain reaction step. Denaturation, annealing, extension. तो यहाँ पे याद रहे हर स्टेप के लिए अलग अलग टेम्परेचर. डिनेचुरेशन के लिए 92 डिग्री सेंटीग्रेड, अनीलिंग के लिए वी हैव टू गो फॉर अराउंड 55 डिग्री सेंटीग्रेड, एक्सटेंशन के लिए द रिक्वायर्ड टेम्परेचर इज 72 डिग्री सेंटीग्रेड. सो दिस इज व्हाट वी आर गोइंग टू डू in vitro DNA synthesis, polymerase chain reaction, amplification of DNA. 3 steps, denaturation, annealing, extension. Fourth option is the correct option. Okay, the next question is select the correct match. तो हमारे पास 4 options हैं, we have to select the correct match. The first option is T.H. Morgan. उसका जो opposite matching है transduction. T.H. Morgan, he has carried out the genetic experiments, not the transduction. The second option is तो इसका मतलब first option is wrong. The second option is F2 into recessive parent with dihybrid cross. Dihybrid cross में it is the cross between the 2 parents having the 2 contrasting characters, और 2 characters के बीच में जब क्रॉस होता है तब हम उसको डायहाइब्रिड कहते हैं. And the third one is ribozyme nucleic acid. Well, this seems to be the correct one, but we will see for the next one. That is the George Mendel. Well, George Mendel, very famous, we know. He has carried out the experiments on garden pea plant. Monohybrid cross, dihybrid cross. तो ट्रांसफॉर्मेशन नो. तो जो बचा हमारा ऑप्शन है वो है राइबोजाइम. तो राइबोजाइम इट इज़ अ न्यूक्लिक एसिड विच हैज़ अ कैटालाइटिक एक्टिविटी. इसका क्या है? कैटालाइटिक एक्टिविटी है and it is a nucleic acid. Usually we say nucleic acid, RNA and DNA. The ribozyme it is also a nucleic acid. Same RNA से बना हुआ है but इसका function क्या है? इसका catalytic activity है. So one more question from biotechnology. So our next question is, a new variety of a rice was patented by a foreign company. Though such varieties have been present in India for a long time. This is related to. Well, this is related to something again biopiracy. ये biopiracy से related है. So in the biopiracy we have seen that biopiracy means it is a legal theft. So American multinational company what they have done, they have collected the Indian rice variety. उन्होंने Indian rice variety को लिया. उस राइस वैरायटी को दे हैव क्रॉस्ड विद द सेमी ड्वार्फ वैरायटी. सेमी ड्वार्फ वैरायटी के साथ क्रॉस किया. बाय दिस दे ओब्टेन अ न्यू वैरायटी. उनको इनसे एक न्यू वैरायटी का राइस मिला. दे गॉट अ न्यू वैरायटी ऑफ अ राइस एंड दैट राइस दे नेम्ड इट एज. So they got a new variety of a rice and they named that new variety of a rice is that is the Basmati, and they have commercialized this rice. So now the question is that what is this related with? A new variety of a rice was patented. As I said, it is an American company who got patent on Basmati obtained from a Indian राइस वैरायटी. तो अगर हम ऑप्शन देखें, लर्मा रोज़ा, शरबती सोनारा, सो ये दोनों दे आर नॉट द राइट राइस. दे आर नॉट द राइस वैरायटी, दे आर वीट वैरायटी. एंड जो राइस वैरायटी बचती है CO667 and Basmati में we know that Basmati it is an Indian rice variety जो बहुत commonly यूज़ किया जाता है. हमारे पास इंडिया में around 27 different varieties of Basmatis are documented. So American company they have collected the rice variety, Indian Basmati rice, crossed with the semi-dwarf variety and they obtain a Basmati, and this is again an example of biopiracy. तो यहाँ पे जो आंसर बैठता है वो है बासमती राइस. Okay, so the next question is, which of the following pairs is wrongly matched? The options are exotype sex determination in grasshopper, ABO blood grouping is codominance, starch synthesis in P, multiple alleles, TH Morgan linkage. Now we'll look for the correct option under this. The XO or X0 type of sex determination is seen in grasshopper. ये सही है. grasshopper में X0 या XO type of sex determination होता है because here the Y chromosome is absent. Right. The next option ABO blood grouping. ABO blood grouping. The codominance का जो example है वो है ABO blood grouping. कोडोमिनेंस मतलब बोथ द एलील्स एक्सप्रेस्ड इक्वली, उसको हम कोडोमिनेंस कहते हैं. तो द बेस्ट एग्जांपल फॉर द कोडोमिनेंस इज एबीओ ब्लड ग्रुपिंग. याद रखिए वी आर गोइंग फॉर द रॉन्ग मैच. तो ये दोनों सही मैच हैं, तो दिस कांट बी द ऑप्शन. द थर्ड वन इज स्टार्च सिंथेसिस इन पी, मल्टीपल एलीज. मल्टीपल एलीज स्टार्च सिंथेसिस? नो. स्टार्च सिंथेसिस is because of a gene जिसको हम कहते हैं Pleiotropic gene. What is this pleiotropic gene? Pleiotropic gene is a gene which is going to control more than one character. वो एक से ज़्यादा कैरेक्टर को अगर कंट्रोल करता है तो हम उसको प्लियोट्रोपिक जीन कहते हैं. फॉर एग्जाम्पल, इफ दिस इज अ जीन, इट इज़ गोइंग टू कंट्रोल अ कैरेक्टर 1, that is the trait 1. It is also going to control one more character, trait 2. It is also going to control one more character, that is trait 3. तो such gene is known as pleiotropic gene. हमने यहाँ पे ऑप्शन देखा है, इट इज़ अ मल्टीपल अलील्स. मल्टीपल अलील्स इज समथिंग डिफरेंट. तो मल्टीपल अलील्स का हमारा एग्जाम्पल अगेन आता है हमारा ABO ब्लड ग्रुपिंग जो है, that is the example for the multiple alleles. So starch synthesis in the pea can't be the multiple alleles. It is a pleiotropic gene. Well, we will look for the fourth option also. T.H. Morgan linkage. Yes, T.H. Morgan has carried out an experiment to show the linkage between the genes and how they are inherited. तो यहाँ पे as per the question, that is the wrongly matched option is the starch synthesis. Starch synthesis by pleiotropic gene, not it is a multiple alleles. So the next question is select the correct statement. The options are spliceosomes take part in translation. Punnett square was developed by a British scientist. Franklin Stahl coined the term linkage. And the fourth option is transduction was discovered by S. Altman. So now coming to the first option, spliceosome take part in translation. Well, translation, it is a process. We know that translation में क्या होता है? Translation में mRNA के ऊपर जो information होता है, that is used for the synthesis of polypeptide chain. वो polypeptide chain में synthesize होता है, उसको हम translation कहते हैं. And in the translation, nowhere the spliceosomes are used. So where the spliceosomes are used? The spliceosomes are used in a process called as post-transcriptional process. Post-transcriptional process में ये spliceosomes use होता है. They are used in post-transcriptional process to remove the introns. Why? To remove introns. So it's nowhere concerned with the translation, right? So this is a wrong option. Next, coming to Punnett square was developed by a British scientist. ये जो Punnett square है, this is usually used by or it is developed by a British scientist named as R.C. Punnett, and he is a British scientist. So this Punnett square, it is used in monohybrid and dihybrid cross में use किया जाता है, and it is developed by, as I said, he is a British scientist. So this seems to be the correct option, but we'll go for the next one. The Franklin Stahl coined the term linkage. No, the Franklin Stahl, the person, he has given the process of semi-conservation, mode of DNA replication. तो DNA replication semi conservative method से होता है. So that was given by Frank Stahl. He has not given the term linkage. And the last one is the transduction was discovered by S. Altman. No, the transduction was discovered by a person named as, so the two persons are involved here. One is the Ginder and the second one is Laderberg. So these were the two persons who has discovered the transduction. तो obviously इन सब में देखा जाए तो जो ऑप्शन सही बैठता है वो है second option, that is the Punnett square was discovered by British scientist and the name of the scientist is R.C. Punnett. It is used for monohybrid and dihybrid cross. The next question is the experimental proof for semi-conservative replication of DNA was first shown in. Options are plant, bacterium, fungus and virus. तो ये जो फर्स्ट एक्सपेरिमेंटल प्रूफ दिया गया था फॉर द रेप्लिकेशन ऑफ डीएनए एंड दैट टू द सेमी कंजर्वेटिव रेप्लिकेशन is shown in bacteria and that too the bacteria that is E. coli. E. coli में semi conservative replication of DNA दिखाया गया था and that was by Michelson and Stars. So these were the two persons who used the E. coli, that is the bacteria, to show the semi conservative replication of DNA. So हमारा जो option है वो है second option, that is the bacterium. Question is which of the following flowers only once in its lifetime? Only once in its lifetime. Okay. The next question is which of the following flowers only once in its lifetime? Usually हम plants को दो ग्रुप में डिवाइड कर सकते हैं based upon the flowering. So one plants are monocarpic. Monocarpic plants वो होते हैं which are going to flower only once in their lifetime, maybe in a year of 50 or 100 years. And another one is polycarpic. तो polycarpic वो plant होते हैं which are going to produce the flowers and fruit every year, more or less every year throughout their lifetime. अब options में देखते हैं, we have a mango, we used to get the mango every year, so this can't be the one. Jackfruit and even the papaya, ये हमें हर साल fruits मिलते हैं, that means the plants are going to flower more or less every year. So the option which is left out is the bamboo species. The bamboo species is polycarpic. It is going to produce the flower only once in its lifetime. That I say that 50 or 100 या 50 या 100 साल में एक ही बार ये plants flowering करते हैं. So the option is the bamboo species. Next question is offsets are produced by. Options are parthenocarpy, mitotic division, meiotic division and parthenogenesis. तो यहाँ पे जो फर्स्ट ऑप्शन दिया गया है दैट इज पार्थेनोकार्पी. पार्थेनोकार्पी ये एक फ्रूट है विच इज डेवलप्ड विथआउट फर्टिलाइजेशन. फर्टिलाइजेशन के बिना जो फ्रूट डेवलप हो रहा है उसको हम पार्थेनोकार्पी कहते हैं. एंड दीज फ्रूट्स आर यूजुअली सीडलेस. ये सीडलेस फ्रूट्स होते हैं. माइटोटिक डिवीजन वी नो इट इज नॉर्मल cell division which usually occur for growth के लिए या development के लिए जो cell divide करते हैं उसको हम क्या कहते हैं? we call it as mitotic division. Meiotic division does not takes place in somatic cell. somatic cell में meiotic division absent होता है. So the meiotic division offset it is a plant. So यहाँ पे हम देखेंगे meiotic division is absent. Now coming to the parthenogenesis. So parthenogenesis it is a process of development of embryo without fertilization. Without fertilization the embryo is developed. उसको हम parthenogenesis, the process of development of embryo without the fertilization. So now which is the correct answer? तो offset it is an hydrophytes जो पानी में grow होते हैं. तो यहाँ पे offsets are produced by, that means the growth and development in the offset is because of mitotic division, because meiotic division does not takes place in the somatic cells, right? So the correct option is mitotic division. Okay, so the next question is the match the following, that is select the correct match. So options are Matthew Messelson and F. Stoll, Pisum sativum, Alfred Hershey and Martha Chase, that is the TMV, Alec Jeffreys, that is Streptococcus pneumoniae, and Francois Jacob and Jacques Monod, that is the lac operon. We'll see which is the correct match. The Messelson and Stoll they have no relation with the Pisum sativum because they have given the model for DNA replication. Model for DNA replication. अभी दो क्वेश्चन ऐसे आए थे जहाँ पे हमने देखा methylsen, that is the methylsen and star. They have given a semi-conservative method of DNA replication. Alfred Hershey and Martha Chase. So these are the persons who have given us an important information that DNA is genetic material. DNA is the genetic material, not the protein. So that information is given by the Hershey and Chase experiment. The next one is Alex Jeffreys. So Alex Jeffreys, these are the persons who have shown a very important contribution in the field of DNA fingerprinting. DNA fingerprinting में उनका contribution रहा है. Now coming to the last option, that is the Jacob and Monod. They are related to the lac operon. They have given the concept of lac operon. So हमारा जो option जो सही है, जो correct match है, वो है fourth option. That is the Francois Jacob and Jacques Monod. They have given the lac operon. Okay, so the next question is, which of the following has proved helpful in preserving pollen as fossils? Options are oil content. The second one is cellulosic intine. The third one is pollen kit. And the fourth one is sporopollenin. So यहाँ पे we will see the first option, that is the oil content. It has nothing to do with the preservation of pollen. So it is a wrong option. Cellulosic intine. Cellulosic intine. It is the inner membrane of पोलन के अंदर जो एक मेम्ब्रेन रहता है जिसको हम यूजुअली वी कॉल इट एज इंटाइन, वी कॉल इट एज इंटाइन मेम्ब्रेन. सो cellulosic membrane. So again, this is also not going to help us in preserving the pollen as fossils. The third one is the pollen kit. Pollen kit, it is used for pollination in insect. ये इंसेक्ट में पॉलिनेशन के लिए यूज किया जाता है. द लास्ट वन इज द sporopollenin. So sporopollenin, it is used for preserving the pollen as fossils. The reason is that it is neither easily degraded by enzyme or acid or alkali. So that's why these are preferred to preserve the pollen as fossils. तो हमारा जो सही ऑप्शन होगा वो होगा fourth option, that is sporopollenin. Our next question is natality refers to. डायरेक्ट क्वेश्चन है बट फिर भी हम लोग ऑप्शन से एक बार देख लेते हैं. The first option is number of individuals living the habitat. The second option is birth rate. Third option is death rate. Number of individual entering the habitat. So number of individual leaving the habitat is called as immigration. Number of individual entering the habitat, immigration. Birth rate is known as natality. Death rate is known as mortality. So the natality refers to birth rate. The number of birth in a particular area at a particular given time is known as natality, and that natality, it is going to represent the birth rate of that population. Okay, so our next question is World Ozone Day is celebrated on. It is a very general question and very simple question. So World Ozone Day is celebrated on 16th September. Okay, the next question is which of the following is a secondary pollutant? तो secondary pollutant के लिए हमारे पास ऑप्शन है SO2, CO2, COO3. So for this we will see this one. So what exactly is the secondary pollutant? Secondary pollutant which is formed from the primary pollutant. For example, here you can see in this diagram, that is the oxides of nitrogen combines with the hydrocarbons, it can be volatile organic compounds, and this in presence of uh, solar radiation, that is the high intensity of solar radiation, high solar radiation. तो इसके presence में you are going to get a secondary pollutant, that is O3, ozone. So ozone is our secondary pollutant. Ozone is represented by O3. So हमारा option जो होगा वो होगा fourth option, that is O3. O3 is a secondary pollutant. The next question is, NEET is the range of temperature that organism needs to live, the physical space where an organism lives, all the biological factors in the organism's environment. So, well, the fourth option is the functional role played by the organism where it lives. तो ये जो first option है, the range of temperature, we can say the optimum temperature which is required. So this is not related to the niche because it is an address of an organism where it lives, where it functions. So this is not related to that one. Physical space where organisms live, physical space, it is an habitat where the organisms are living. So that is also not related to the need. All the biological factors in the organism's environment, ये भी नहीं, but इसका जो सही option बैठा है, the functional role played by the organism where it lives, a place where the organism is living and the activities or the function which the organism carries is known as the need. So हमारा जो सही option है वो है fourth option. The next question is, what type of ecological pyramid would be obtained with the following data? तो यहाँ पे जो डाटा दिया गया है, that is the secondary consumer 120 ग्राम, primary consumer 60 ग्राम, primary producer 100 ग्राम. तो जब हम ग्राम बोलते हैं तो that means it is representing the biomass. Biomass को हम either we can consider the in the form of, not either, it is considered in the form of weight. weight के form में consider कर रहे हैं, तो इसका जो unit होगा वो होगा ग्राम. So when you are going to draw the pyramid for this one, तो pyramid में हमेशा the base of the pyramid is for producer, that is the P, and here the producers में जो biomass है वो कितना है, that is 10 gram. The second level they are saying that the primary consumer का जो biomass है, it is more than the primary producer, that is 60 gram. These are the primary consumer, this is the primary producer will be. And the third which is given, that is the secondary consumer. तो secondary consumer में biomass और भी ज़्यादा है, that is 120 gram. So as a result of this, we are getting a pyramid of inverted shape. So here we are getting an inverted pyramid. So now we'll look for the options here. The first option is upright pyramid of number. जैसे मैंने कहा, here the ग्राम में हमें यहाँ पे दिया गया है whatever, like, so it is not in the numbers, so obviously it should be a biomass, तो ये option नहीं हो सकता. The second one is pyramid of energy. जब हम energy को measure करते हैं, we are going to take the kilocalorie. This is the unit for the energy, so this can't be the option. The third option is inverted pyramid of biomass, and the fourth one is upright pyramid of biomass. इन दोनों में से हो सकता है, but when we draw the pyramid we are getting an inverted shape, तो उसका जो option होगा वो होगा third one, that is inverted pyramid of biomass. So the answer here is the third option.